Director of the School of Global Environmental Sustainability at Colorado State University, and the chairperson of this National Academies Committee. In my role as chair of this study and as the moderator of this committee meeting, I'd like to welcome everybody here who's on Zoom, those of you watching the live stream, and all who may watch this recording later. The task being undertaken by this committee of the National Academies of Science, Engineering, and Medicine is to explore the linkages between soil health and human health. The committee's full statement of task and our challenge can be found on the study website, which is available at the link under project on our website to the right of the embedded video. I would like to give you all a format, just an idea of the format that's coming up. I'm doing the welcome now with some little housekeeping things, and then I will turn it over to Carol Laney, and then we will have the invited presentations. Let's see. Following her presentation, I will introduce the speakers for today. We will adjourn this meeting at 5 o'clock Eastern time, or when the conversation is complete, whichever comes first. To start, I want to note that this is an open, on-the-record session. Interested in individual members of the press have been invited to join as observers. This is not a news conference, however, and we will not be entertaining questions from the audience. Reporters who would like to talk with members of the committee are kindly asked to touch base with the staff officer, Carol Laney. Also, anyone who would like to submit a written comment or document to the committee is welcome to do so by clicking the Provide Feedback link on the right side of this study website. You can also subscribe to updates on this committee and its progress by signing up for the National Academy's Board on Agriculture and Natural Resources listserv, also on the right side of the webpage. I would also like to remind everybody that this is an information gathering session. That is, the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and recommendations. Therefore, I ask everyone to be extremely mindful of the fact that the committee has made no conclusions yet, and that it would be a mistake for anyone to leave the meeting today thinking otherwise. Comments made by individuals, including members of the committee, should not be interpreted as positions of the committee or the academies. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will deliberate thoroughly before writing its draft report. Moreover, once the draft report is written, it must go through a rigorous review by experts who are anonymous to the committee, and the committee must then respond to this review with appropriate revisions that adequately satisfy the Academy's Report Review Committee and the NAS President before it is considered an official Academy's report. This committee is comprised of 15 volunteer experts. Their names are listed there for you, and they're also. I'd now like to turn things over to Carol Laney, a senior program officer with the Board on Agriculture and Natural Resources at the National Academies and the study director for this particular activity. Carol will give you a brief overview of the National Academy's study process. Carol?
Thanks, Diana. I just want to give a brief overview of the National Academies in general and the study process specifically for those of you who joining us today who might not be familiar with the institution. The National Academy of Sciences was established by a bill passed by Congress and signed into law by President Lincoln in 1863. Later, the Academy was granted the authority by executive order to establish the National Academy of Engineering in 1964, and later the Institute of Medicine, which became the National Academy of Medicine in 2015. Today we are the National Academies of Sciences, Engineering, and Medicine, and so the organization consists of the 3 honorific bodies, as well as the work of scientists across the country, both in and outside the academies, who provide expert advice to the nation on issues of science, engineering, and medicine. The mission detailed in the Congressional Charter is to investigate, examine, experiment, and report upon any subject of science or art whatsoever when called upon by the government. We fulfill this mission through many convening activities, including consensus studies like the one which we are discussing today. Just to share a little bit more detail about consensus studies, as opposed to other convening activities we organize, such as workshops and forums, consensus studies are conducted by committees convened specifically for a unique statement of task. The experts who serve on these committees do so as individuals, not as representatives of any organization or institution to which they may belong. They also serve as volunteers. Consensus study members review the state of scientific literature to write a report that responds to the unique statement of task, and that report will hopefully be used to shape sound policies, inform public opinion, and advance the pursuit of scientific field in question. Each consensus study goes through a number of stages. First, the statement of task is defined through conversations between the national academies and the study sponsor. The study sponsor for this particular committee is the USDA's National Institute of Food and Agriculture. Next, the National Academies asks for nominations to identify individuals with the necessary expertise to address the statement of task. Each committee should be comprised of individuals with the appropriate range of experience and contain a balance of perspectives. Nominees are appointed to the committee by the President of the National Academy of Sciences, Dr. Marcia McNutt, and their committee membership is provisional until they have completed a discussion of conflict of interest, disclosures, and composition at their first meeting, which this committee wrapped up last month. After being appointed, the committee moves into its information gathering stage and holds meetings like the one we are doing today, where it invites in people to speak to them in person or via Zoom. Members of the public may also submit information to the committee or documents that they may want the committee to consider as it addresses its statement of task. The committee will soon begin to deliberate about how to best address the statement of task and then draft a report. As Diana mentioned, when that draft report is complete, the document is reviewed by anonymous reviewers, and the committee must revise the report in response to those comments. The National Academy's Report Review Committee determines when the reviewers' comments have been adequately addressed and approves the report for release. The report is then made available to the sponsor as well as to the public. I just want to say a few more words about the committee meeting process. When the committee hears from speakers, such as we are doing today, it meets in what we call open session. Anyone from the public may watch the presentations, and if we meet in person, which we did in April, and we have 2 more in-person meetings coming up this summer, then people can attend in person as well. When the committee meets in closed session, it does so to deliberate on the statement of task and devise the best way to address it. And when they meet in closed session, only the committee members and staff are present. So this is the general timeline for this study. We've already gone through committee formation, obviously, and we're in the information gathering phase. This is our 3rd meeting, and we have several more planned this summer in June and July, including 2, as I mentioned, that will be in person. I anticipate that the draft report will be ready to be reviewed in the late fall, and that the final report will be released in the spring of 2024. You can see this committee statement of task here, and I'll just pause for a moment to read it. And Eric, if you go to the next slide, here's the remainder of the statement of task. As I mentioned, USDA's National Institute of Food and Agriculture is the sponsor for this study. And it came to us via NIFA in a congressional request in last year's appropriations legislation. So finally, here's a screenshot of the study's website. You can see on the left tabs to find the full statement of task, the committee names and bios, and a list of our past recordings of our past events. On the right, in the oval, you see the button you can click to provide feedback to the committee, and below is the box where you can subscribe for updates about the study. And if you would wish to contact myself or Kat Keynes, who is supporting this study, our email addresses are there. And also, we will post information about upcoming meetings when the agendas are available for those that later this summer. So with that, I'd now like to pass it back to Diana.
Hi, everyone again. Thanks, Kara. It's now my pleasure to introduce the first of our invited presentations today. Jamie DeWitt is a professor in the Department of Pharmacology and Toxicology of the Brody School of Medicine at East Carolina University. She has PhD degrees in environmental science and neuroscience from Indiana University, Bloomington. A major emphasis of Dr. DeWitt's current research program is on the effects of pure and polyfluoroalkyl alkyl substances known as PFAS. She has numerous publications on the toxicity, has served as an external reviewer of PFAS documents for various agencies and entities, and has served and does serve as plaintiff expert witness in cases involving PFAS. Jamie, thanks so much for joining us today. I look forward to your presentation.
Well, thank you very much, Diana, and thank you to the committee membership and the staff for inviting me here today to talk to you all about PFAS. You're probably wondering a little bit about PFAS and soil microbe health. I'm not going to talk about that. I'm just going to give you a high-level overview of what PFAS are and what human health concerns have been associated with their exposure. Dr. Wall already shared some of my activities, but I just wanted to share with you some of my declarations. I am funded to study the toxicological effects of PFAS. I receive funding from the state and federal government. I was a member of the EPA PFAS Science Advisory Board that has made some recommendations regarding regulatory limits for certain PFAS in drinking water. I speak about PFAS a lot, and as Dr. Wall said, I do serve and have served as a plaintiff's expert witness in cases involving PFAS. Well, I just wanted to start with, as I mentioned, a high-level overview of what PFAS are and why we should be concerned about them. PFAS is an acronym for per and polyfluoroalkyl substances, and this is a large class of synthetic chemical compounds that contains, to date, maybe over 10 or 12,000 individual compounds. From a toxicological perspective, this makes PFAS very challenging to study because there are so many individual compounds. As you can see here in this figure, they can be broken down into different subclasses, and these different subclasses are based on the number of carbons there are in the compounds, as well as the different functional groups that are affixed to the carbons, as well as their final designations in terms of whether they're used as polymers or as surfactants or as starting agents for some of the fluoropolymers. We know the most about PFAS that we refer to as legacy or long chain PFAS, as you can see here. I've highlighted two long chain PFAS. These are perfluoroalkyl acids, and there will be a quiz at the end to see if you remember all of the acronyms. The two, as I mentioned, that we know the most about are perfluorooctanoic acid, or PFOA, or perfluorooctane sulfonic acid, or PFOS. As I mentioned, these are considered to be legacy long chain, and PFOA and PFOS are part of a proposed maximum contaminant level guideline that the US EPA has recently issued. But we also have some novel or understudied or alternative or replacement or short chain PFAS that have emerged into the environment and that also have concerns associated with them. Some of these include chemicals that are part of a process known as Gen X chemistries. The compound that I have here is actually HFPODA, which is referred to colloquially as Gen X. You can see it has fewer numbers of carbons, but there's an oxygen inserted into the chain of carbons. This makes this a perfluoral ether acid. Again, more acronyms. PFBS is an example of a short chain perfluoroalkyl acid that doesn't have an oxygen in it. These two chemicals also are part of some proposed regulations by the US EPA. PFAS are in many different products and processes. If you look at the picture here, you can see that they are involved in the production of or are direct products in many items that we might have in our work environments or even our home environments. They're used predominantly for their stain, oil, and water resistant, as well as their non-stick properties. They are extremely durable, they are extremely stable, which is why they've become so widely used in many industrial products and processes. But as you will see, this stability and durability is one of the reasons that they have become concerns for human and environmental health. PFAS can get into the environment through many different pathways. That is one of their concerns. They can be emitted. They can be emitted during production. They can be emitted during their use phases, and they can be emitted at the end of a product's use phase. So they can get into the environment. We also are concerned with PFAS because of their persistence. I mentioned that they're very durable. A lot of the durability comes from the strength of the carbon-fluorine bond, which means that PFAS are among the most long-lived persistent organic pollutants that we know of today. It's estimated that the natural degradation of PFAS may be on the orders of hundreds or even thousands of years, although hopefully we can take advantage of some microbial activity to speed up the degradation of PFAS. And hopefully Dr. Lee and Dr. Sharp will talk a little bit about those processes. We're also concerned about PFAS because many of the ones that we've studied can move from the environment into the bodies of living organisms, including humans. So if they can move from the soil into plants where they can be ingested, they can move from drinking water into bodies of people who consume that contaminated drinking water. PFAS, unlike a lot of other persistent organic pollutants, also are highly mobile in the environment. They have both lipophilic and hydrophilic properties, which means that they can exist in many different compartments of the environment, and they can move to points distant from the sites of their release, which makes them even more challenging to manage because of their ability to move from place to place. The rest of my presentation will focus on their toxicity and specifically on the information that many epidemiological studies have given to the scientific community about what PFAS may do to human health in terms of disease risk. Before I get into that, I just wanted to share a little bit about what we understand about PFAS exposure to humans. This is a figure from a paper that is slightly dated, but it is still relevant today. In this paper, Dr. Vestergren and Dr. Cousins looked at an average human male and predicted what his exposure would be in terms of where he would get exposed to PFOA, given different areas that he might encounter PFOA. And I want you to take a couple of things away from this figure. If you look at figures A and B, you can see that for somebody who's got slightly elevated concentrations of PFOA in their drinking water, or who lives in an area where their drinking water doesn't have much PFAS in it, the majority of this person's exposure comes from their diet. And dietary exposure can be from food packaging in which contain PFAS, but dietary exposure can also come from the uptake of PFAS from the soil environment into foods that somebody might grow at home, purchase from a farmer's market, or purchase from a grocery store. If you look at figure C, you can see that if somebody lives in an area with a known point source of PFOA contamination to their drinking water, the majority of their exposure will come from that drinking water exposure. So for most people who don't live in PFAS contaminated areas, dietary routes of exposure are going to be the major source of their PFAS exposure. I'm going to talk now about what we know of the toxicity of PFAS and human health risks, and I wanted to highlight that the majority of information that we have comes from just a handful of the 12,000 PFAS that are thought to exist. I'll mostly be talking about PFOA and PFOS, not double PFOA, PFHXS, PFBS, PFNA, and Gen X chemicals. There have been linkages made to other PFAS chemistries, but these are the ones about which we have the most information. One of the health risks that has been associated with exposure to these PFAS is elevated serum cholesterol. This is a problem because it can increase the risk of various cardiovascular diseases, including heart disease and stroke. Another type of adverse health outcome that I study in my laboratory is immunosuppression, and this has been measured in people exposed to PFAS through a diminished response to vaccines. This has mostly been observed in studies where children have been evaluated, and they have an attenuated response to their childhood vaccines. But what this tells us is that when somebody has a reduced response to a vaccine, other parts of their immune system may be impaired, which increases risk to various types of infections. It also increases risk to cancers because the immune system is very important in helping to prevent cancers from developing in our bodies. Developmental toxicity is an outcome that has been linked with PFAS exposure as well. One of these is low birth weight. There's also been associations of pregnancy-induced hypertension and preeclampsia, which put the mother and the baby at risk. We know that children who are born with low birth weights are more likely to develop health problems later in their lives. Increases in serum liver enzymes have been linked to PFAS exposure. These are signs of liver damage and liver disease. Animal studies also demonstrate that the liver is a very sensitive target organ of PFAS exposure. Animals experience something known as hepatomegaly, or enlarged liver, due in part to increases in liver enzymes associated with beta fatty acid oxidation. So liver damage is also an outcome that has been linked to PFAS exposure. There's also been several types of cancer linked to PFAS exposure. For both PFOA and PFOS, we see evidence of different types of tumors in studies of experimental animals. For PFOA, we have linkages to human studies or to human epidemiological studies tying kidney cancer and testicular cancer to their exposure. But as you can see here, multiple organs have been implicated as being sites of tumor cell development from PFAS exposure. I also wanted to highlight that many individual chemical studies of individual PFAS in experimental animal models have shown concordance with findings in epidemiological studies, which increases our confidence in the epidemiological findings from the various studies that have been conducted. So when we see this level of concordance, our level of uncertainty diminishes. I'm highlighting here a really great figure from the most recent maximum contaminant level proposed document put forth by the United States Environmental Protection Agency. The proposed maximum contaminant level for PFOA in drinking water is 4 parts per trillion. This is an extremely low level, and it is probably one of the lowest MCLs recommended by the US EPA to date. This MCL was based on a reference dose that represents low dose concordance among 3 of the specific endpoints that I just explained. This would be decreased vaccine responses, impacts on developing organisms, and impacts on serum cholesterol. So these 3 different endpoints occur at about the same dose, leading to a reference dose that is extremely low. That's 3 times 10 to the negative 8 milligrams per kilogram per day. And this was used by the US EPA to derive the maximum contaminant level in drinking water of 4 parts per trillion. I also wanted to point out here that all of these different studies for immune effects, developmental effects, and cholesterol effects are from epidemiological studies. So this endpoint is based on findings in humans that have been exposed to PFOA. If we look at the maximum contaminant level proposed by the US EPA for PFOS, it also is 4 parts per trillion. It's based on low birth weight and increased total cholesterol. The doses, or the reference doses that were derived for the immune endpoint, was not quite as low for PFOS as it was for PFOA. So the US EPA selected low birth weight and the cholesterol endpoint for deriving their maximum contaminant level. Again, these are based on human studies. So these studies demonstrate that these health risks occur at very low concentrations of PFOA and PFOS in drinking water. The US EPA, in their proposed maximum contaminant levels, also recommended maximum contaminant level goals for carcinogenic risks of PFOA and PFOS. In previous documents, the US EPA indicated that the data were sufficient for PFOA and PFOS to be classified as suggestive human carcinogens, but in re-evaluating the data and evaluating some of the newer data that had come out in its most recent recommendations, the US EPA upgraded or from a health perspective, downgraded PFOA and PFOS to likely to be carcinogenic for humans. As you can see for PFOA, this was based on evidence of kidney and testicular cancer in humans, and then a range of different cell tumor types in experimental animal models. For PFOS, this designation was made based on evidence in experimental animals. So both PFOA and PFOS are considered by the US EPA to be likely to be carcinogenic to humans. The next designation is human carcinogen. So this is one step below an agent that would be classified as a known human carcinogen by the US EPA designation for carcinogenic chemicals. I also wanted to highlight the findings of the other 4 compounds that are part of the US EPA's proposed maximum contaminant levels. These 4 compounds are GenX, PFNA, PFBS, and PFHXS. So just to remind you, GenX and PFBS are short chain compounds. PFNA and PFHXS are considered to be long chain compounds. For GenX, and I also would like to point out that for these particular compounds, the reference doses are based on experimental animal evidence. GenX has a critical effect of liver effects. These are actually effects observed in dams. These are female mice that were dosed with GenX and then were bred with male mice to produce offspring to look at developmental effects. These female mice had signs of toxicity in their liver, which the US EPA used to derive their reference dose for GenX. PFNA reported is based on reports of developmental effects in mouse offspring. These have to do with delays in development. PFBS and PFHXS are based on endpoints in experimental animal models that reported changes in thyroid hormone levels. So these would be considered to be endocrine disrupting effects. So you can see here that for these 4 compounds, there appear to be different health effects, but what's important from this particular maximum contaminant level goal recommendation is that the US EPA is using something known as a hazard index approach. This is a somewhat complicated approach. I won't go into the details of it now, but the idea behind it is that these 4 chemicals can act in synergy to increase the risks of the individual health effects exhibited by just one of the chemicals. So when these chemicals are together in a mixture, as they often are in the soil environment or in the water environment, the risks of these different health effects increases because of the mixture. I wanted to highlight a report that the National Academy, a committee of the National Academies submitted or published in late 2022. This was a report that was assigned to evaluate epidemiological evidence and to make recommendations for clinicians about potential increases in standards of care that they should apply to patients living in areas of high PFAS water contamination. The committee looked at 7 different PFAS and looked at epidemiological information and found that for these 7 PFAS, there was sufficient evidence of an association for immune effects, cholesterol effects, cancer effects, and effects on cholesterol. There was limited suggested evidence for other types of health outcomes, and this committee made recommendations about what healthcare providers could do for patients with concentrations of these 7 PFAS in their blood. So a patient could have their blood measured, get information on these 7 PFAS, and work with their healthcare providers for figuring out if they have any signs or symptoms of these particular health risks. So this is a great tool that patients have to kind of take command over their own health if they are concerned about PFAS in their communities. I also wanted to speak just a little bit more about the recommended maximum contaminant level goals that the US EPA has issued. What's important to consider about these goals is that it's 6 PFAS, which is a very small fraction of the total amount of PFAS out there. But what's really important is that when the US EPA looked at the technological feasibility of implementing these MCLs, they determined that with the technologies that we have available today for drinking water treatment, treating for these 6 PFAS would likely attenuate levels of other PFAS in drinking water. So although this is only 6 PFAS, there is hope that communities struggling with PFAS in their drinking water would have many other PFAS removed with the technologies that would have to be implemented to meet this maximum contaminant level if it is actually promulgated into a regulatory standard. And with that, I'm finished a little bit early and I'm happy to take any questions that you all may have.
Thank you very much, Dr. DeWitt. That was really informative as we were hoping for, but the update is kind of scary. I'm glad to see the end point on the recommendations. Yeah, we're now open for questions. But does anybody from the committee have questions to start with? Yeah, Mike Rusek.
Yeah, thank you. I was trying to find the raise hand, but I couldn't find it. So thanks so much for that very nice summary. A question about the proposed MCLs on PFAS or on PFOA and PFAS. Can we measure levels that low?
I believe at this point we can measure down to 4 parts per trillion. In the initial recommendation, the health advisory levels were below limits of detection, but at this point we can get down to about 4 parts per trillion, at least for PFOA and PFOS. Some of the other compounds, not so much. Part of that is because we don't have standards for all of the other compounds, so we can't quantify levels of some compounds that exist in the environment right now.
Okay, thank you.
Rebecca Nielsen on the committee.
Yeah, thank you. Yeah, thanks so much for that excellent presentation. So I was hearing that if total organic fluorine is mentioned, so just the amount of that CF bond, that you kind of can sum up across those however many thousand, I keep hearing different numbers for how many thousands of PFAS particular species there may be. And then that, you get like an estimate that's, 10 or 100 times more. And will that then help or hinder the clarification of these things? So it seems to me there's a couple issues. One is the extreme expense of getting the data to be able to understand and monitor the problem. And then this sort of issue about the invisibility of most of the species because you can't test for them. So do you have any comment that you might offer us about how things might change as if we can mention this total organic fluorine instead of specific species?
I think that might be a question that Dr. Lee or Dr. Starp might be better able to answer if they could jump in. I know that there is a lot of debate within the scientific community about what approaches should be used for measuring PFAS. Should it be total organic fluorine? Should it be individual species? Is there another type of mass balance approach that would give us that information? I don't know if I'm the best person to answer that question, though.
So do you want anybody to chime in now?
Sure.
I think total fluorine, gives us a picture of something, but there's a lot of things that we don't call PFAS that can be in biosolids that include pharmaceuticals, antimicrobials, pesticides. And so I think when we're doing total fluorine, we have to be careful. And then all PFAS are not equal. And I would hypothesize to say that a large amount of PFAS that might be in biosolids that we're not quantifying are likely side chain polymers that will have very, very slow degradation. And I'll talk a little more about that to the PFASs that we quantify, the PFASs that can get into our water supply. So I feel like total fluorine would not be a fair measure in terms of trying to make calls about what to do.
Yeah,
but they definitely give us a snapshot of something.
And Dr. Lee brings up a very important point, and that is several different definitions of PFAS exist. The US EPA has a definition, OECD has a definition, World Health Organization has a definition, and some of those definitions intentionally exclude certain types of compounds. I can't say definitively, but my understanding of the US EPA definition is that compounds that could be classified as pharmaceuticals and compounds that could be classified as pesticidal substances were largely ignored or not included in the definition because they would be managed by other agencies. So there is ongoing debate within the PFAS community about the proper definition for all PFAS.
Thank you, Jamie. We have one more. Well, we have two more questions, and I think we've got time to take them. Katrina and then Nick.
Thank you for that wonderful presentation. I was curious if you were able to speak a little bit more about the modes of exposure. So it looked like those pie charts at the beginning you showed were primarily food and water. There was one that looked like there was a lot of air involved, and very small slivers for the maybe percutaneous exposures from different types of things people may be putting on their body. Do we know whether do we know much about the mode of exposure, and whether it's primarily gut mediated for food and soil, or if it could also be, for food and water, or if it can also just be actual exposure being near soils that could impact the exposures?
Yeah, from that particular figure, the figure that showed mostly inhalation exposures was for an occupational setting. So we know that people who work in floral manufacturing facilities get their exposures mostly through inhalation of the material as it's being developed or mixed or processed in some way. So for the average person, for me, for example, I don't live in an area where there's a high amount of known PFAS into the water. The major source of my exposure is probably going to be through my diet, through what I ingest, what I purchase at the grocery store for my food, or what I might purchase that has packaging that contains PFAS. But we also know that there can be inhalation exposures through household dust. Some work by Heather Stapleton at Duke University has demonstrated that household dust and different occupational settings contain PFAS, and that's from degradation of PFAS containing materials. So if you live in a home where you have stain-resistant carpeting and stain-resistant furniture and nonstick pans and lots of outdoor equipment that contains PFAS, you're likely going to inhale and possibly dermally absorb some of those PFAS as they become part of the dust in your home. In terms of efficiency by which PFAS are absorbed across different membranes, we know that PFAS are absorbed very easily across the intestinal barrier when they're ingested with food or water. I know there's been at least one published study that looked at the absorption of PFAS in different food matrices. I don't remember the outcome of that. I think the authors looked at different types of meats and how quickly PFAS would absorb across the gut in different types of meat matrices. But when people consume drinking water containing PFAS, the PFAS are very readily absorbed across the intestinal barrier. And without rambling on too much longer, there have been very few studies of lung exposure and of dermal exposure. We do know that PFAS can cross human skin, but not as efficiently as it would cross rodent or pig skin. And we do know that PFAS can be absorbed across the lung barrier, but the number of studies is quite low compared to the number of intestinal or ingestion studies.
Thank you, Jamie and Katrina. Let's take one more question from Nick.
Yes, hi, great presentation. I am interested in the toxicokinetics a little bit. So I'm sure I'm exposed to more than 4 parts per trillion every day in water. And so I have this steady exposure coming into my body. What's the elimination, the metabolism? Is it accumulating in organs? Is the 99.7% of us have PFAS in our blood? What is that like? Is it stable? Can you comment on those things?
Sure. I mentioned that PFAS are persistent. They're persistent in the environment and they're persistent in our bodies as well. PFOA and PFOS have a human half-life of somewhere between 2 and 5 years. So if you eat a gram today, which would be a lot, then between 2 and 5 years from now, you would have half a gram left in your body. And that's if you don't have any other exposures. So this is one of the features of PFAS that makes them concerning. They're slow elimination half-life in people. We also know that PFAS, because they're both hydrophilic and lipophilic, don't really compartmentalize in fat like many other persistent organic pollutants do. They actually hang around in your blood, and they like to stick to proteins. We know that PFOA, for example, sticks to albumin, and that's one of the hypotheses behind why it might be an endocrine disrupting compound, because it hangs on to the protein that is supposed to transport hormones around the body. There's been at least one autopsy study that has looked at where PFAS go, and it looks like they can go into almost any organ, but they're going to be in those organs that are highly perfused or that receive a lot of blood flow because of how they stick to proteins by traveling around the body. Most PFAS we know are excreted in the urine, but they're excreted slowly. So if you're consistently exposed, then you're going to build up levels of PFAS in your blood, and that will give time for PFAS to interact with other molecules in your body that can lead to those health risks.
Thank you very much, Jamie, for your talk and for the questions. We're now going to move on to our next speaker. Our next speaker is Linda Lee, who you've already heard from in this discussion. Linda is a professor at Purdue University, Department of Agronomy, and program head for the Ecological Sciences and Engineering Interdisciplinary Graduate Program. She's a faculty affiliate in the Division of Environmental Ecological Engineering. Her research focuses on understanding the processes that govern the environmental fate and remediation of contaminants in various media for use in contamination mitigation, decision tools, and management guidelines in industrial and agricultural settings. Dr. Lee has more than 15 years of experience conducting PFAS research across a range of PFAS subclasses, as we've just seen in this previous presentation, how many there are, with numerous PFAS-specific publications spanning PFAS solubility, absorption, biotransformation, ecotoxicity, remediation, and occurrence in soils, various aquatic matrices, water, biosolids, and biota. So she's looked at a variety. Linda, welcome. Thank you so much for being on the committee and giving us a speech.
Thank you, Diane. Are you seeing there we go.
Yeah.
Thank you very much. And thank you, Jamie. That was great presentation, trying to capture a lot of data in a few minutes. So my job today is talk about PFAS and biosolids. So I'm going to give a little bit of background on PFAS and biosolids, and then we will look at some transformation and release from biosolids. And then I have a couple slides to just show what we're seeing in water supplies, just to keep everything in context, and then a path, my perspective, path forward. So we already know about the issue of PFAS, but I think it's really important to remind ourselves that a lot of what's driving concerns about land application of biosolids and PFAS being present in biosolids is these really low concentrations that we have to try to, maintain levels below that in our water supplies. So when we're talking about PFAS, I really like to remind people that the water resource recovery facilities that create the biosolids that we land apply, because of their value, it's just a conduit of all of our wastes. And I just really like to bring this up because whether we're dealing with the back end or the front end is important. And so when things go through the wastewater recovery facilities, PFAS in general are just going to the effluent or to the biosolids, either in the same form they came in or in a different form due to biological transformation. You might get a little bit lost to volatilization, but dominantly, you're not really able to treat the PFAS in our typical water resource recovery facilities. And so that's just that ends up being our challenge, and it's why they're besides the health effects that Jamie talked about, it's just the fact that these are so persistent that they're hard to address. And I feel like it would be remiss not to just put in context the value that biosolids do serve us, because again, working where our efforts should be on the back end or the front end. And so we know, you talk to farmers that use biosolids, they love biosolids, both because they're affordable, but because they have a long-term impact that synthetic fertilizers don't have. We can see fields, even in our agronomy farm, that had really high applications of biosolids back in the days when they were worried about metals and biosolids, which we found a way to deal with that without just banning land application of biosolids. And you can still see there the crops that come up first and are tallest are still in that same exact area, 40 years later. So they really have a lot of value. And I always feel like we have to keep that in context, because we always see the thing that we're talking about the most, which is the elephant in the room, the PFAS. And we're not the only country, right, that's dealing with managing of biosolids. And I think it's interesting in perspective to know that there's still several countries that land apply a lot of their biosolids, several countries that have always incinerated, several countries that primarily just do landfilling. But obviously here in the US, the latest update that US EPA showed was about 43% is land applied, which is down about 30% from this time frame that I'm showing here for European standards, and about 25% of the total biosolids produced are used in agriculture. But reclamation is also important, but it's pretty small. And then a lot is used in home gardens, landscaping, golf courses. And I think it's important to know that the biosolids that are bagged and sold in the store that people are paying for kind of come under a different scrutiny, but they're really just the same biosolids. So are forever chemicals, and John will talk more about, well, are they forever? Where are we on that measure of forever? And so, like Jamie mentioned, there's lots of subclasses, and really, even for a chemist like me and John, it's just alphabet soup, because there's so many. And back here, just in 2017, we talked about 3,000, and it went to 5 and 7 and 9, and latest EPA wants to say 12,000, although I think some of those are double counting different structure names. But again, the highlight is, they really don't degrade, or they degrade to other PFAS for the most part. And so that's our challenge. And I set that precedence because I that's important to understand when we're thinking about what happens in the treatment plant, what happens after land application. It's all very much similar processes, and it's all just dependent on whether things are aerobic or not, and what nutrients are present to enhance microbial transformation of PFAS, for the most part, to other PFAS. And just so you can see, this is a summary of a snapshot of PFAS that were in biosolids for the last 2018 to 2022. We're now, almost halfway through 2023. And there's you can think about 3 categories for PFAS. You can think about the ones we know about the most. Jamie mentioned them: PFOS, PFHXA, PFOA. Those are really, they can be present initially, but they're also terminal metabolites for what we call precursors. And in between these terminal metabolites, which we tend to call perfluoroalkyl acids, you have lots of intermediates that serve also as precursors to these acids. But then you have a lot of other precursors that people really aren't measuring at the commercial labs. And it turns out that these precursors can be at least 75% of what you're quantifying in the PFAS portion of the fluorine-mole balance that we can actually quantify. And so when we're talking about these DIPAPs, which are very common in paper packaging, coatings on paper, even coatings on textiles and clothing, you're looking at a compound that has a polar functional group with these 2 tails. So every one of these can create at least 2 additional PFAS. So it looks like we're creating PFAS, but we're not really creating them. We're just transforming. And this is kind of my schematic for that. So we have telomere-based PFASs. They tend to form the carboxylic acids. Then we have our electrochemical process PFASs, like what 3M created, and they tend to create our sulfonic acids like PFOS that we're familiar with. And this happens under multiple steps, pathways, and rates. So not very easy to predict. And the problem is they always end up being these terminal metabolites that are still PFAS, and they tend to be more mobile and more soluble. And so if you're looking at, this is a snapshot of what happens in the anaerobic digestion process, which that dominates in most of our wastewater treatment plants right now. What you see is this is what's coming in and this is what's going out. And you can see that this whole blue are those precursors that we were talking about. So we can think about these acids are those terminal end products. These are intermediates, so this gray. And then all these are precursors, things that can lead to these terminal compounds. So you can see just in an anaerobic process that you go from this large portion of precursors to a small portion. And then this is what most commercial facilities are not measuring. They might measure this yellow and this green, but not this blue. And so it looks like, again, we're creating PFAS in the process, but we're not. And if it doesn't happen in the process, it's going to happen when we land apply. And I just was highlighting here as an example, basically all the perfluoroalkyl acids that we think about, they can appear to increase through the treatment process because of precursor transformation. And then we have these pretty stable intermediates that will also eventually degrade to these short chain acids, but they're just evident of biological microbial transformation. And I'm not going to show aerobic data because I've already showing you a lot of data, but aerobically we see a lot more. Aerobic processes at the wastewater treatment plant will lead to a lot more generation of these shorter chain compounds from precursor transformation. Oh, I meant to delete it. It's too much to see. So what happens after land application? That was really my charge, but I felt like you needed to understand that the same processes that are happening in the plant are the same processes that are happening once they're land applied. So first when I think about land application in our agricultural operations, I just want to remind us there's a lot of other PFAS sources in agricultural operations, not because agriculture created them, but because they're being used in products. So they can be in any product that comes out of plastic bottles. We can also have them in oils and lubricants. We can have them in rainwater. We all now know that just about any rainwater sample you take, you're going to find PFAS in there. This is really important because it's raining down on all our agricultural fields. And we don't want to misinterpret, where all of our PFAS are coming from. You're applying biosolids once, rainwater's raining all year long. And so then once we do land apply, what do we care about? Usually accumulation and transformation, what's happening there because that affects mobility. We were concerned about crop uptake because that then gets it into the food chain. We care about leaching because it gets into the groundwater that might impact our drinking water. We care about runoff and tile drain discharge that people don't think about if they don't live in the Midwest. And then that's affecting our biota in our aquatic species and wildlife, but also several regions will use surface water for their drinking water source. So I was going to take this out, but let's just say out of the plant studies, I think it's also important to remind ourselves because, Jamie talked about drinking water and digestion being, our biggest source of PFAS if we're not near a heavily contaminated site. And even for crops, most of the PFAS are the shorter chain PFAS that are going into the crops. A lot of these are not even in the space of being regulated. So that's just important for us to remember. What we don't know is about the precursors and their long-term source to what will stay in the root zone over time. Okay, I already showed you this, so let's get past that. Okay, that was just to remind me. Now I'm going to show you what happens at 3 different scales. I'm going to show you what happens once things are land applied. So this is not a field study. I'm privileged to show you a little data from one field study, but this was an outside mesocosm study that we're funded, where we mixed biosolids in soil, and this was soil that was intentionally chosen to not be highly retentive of PFAS. It was a very sandy type soil, because the goal of this was what happens, what kind of PFAS get released from biosolids under normal environmental conditions and rain events. And so that's what we're going to see here. So I'm going to show you two types of data. First, this is what's leaching. So you can see this is parts per trillion on your y-axis. This is just for the carboxylic acids. So we see our 4, C5, C6, C7, C8, C9. And you can see this is pore volumes is the amount of liquid that's flowing through this column based on how much it can hold. This is over a 6-month period in Seattle. And so you can see, you clearly see things increase and you see these different changes. Now, what's important is this right graph. This is relative to initial, meaning this is what we saw in the leachate. But now if we compare, what did we measure in the initial biosolid that we mixed at 1 to 3% in the soil level? We did this with 7 different biosolids. I'm just showing you from 3 facilities. What you can see is this dotted line is ratio of 1. And what you see is in most of these cases, you have lots of leachate above what you would have predicted if you only considered what was in the biosolid to begin with. And that's because of precursor transformation. You can see the same thing with the sulfonic acids. You see, again, you get your leach concentrations. You'll actually notice that they tend to be lower than the carboxylic acids. And then if you look to relative to initial, you see in a lot, I'm just showing you a snapshot, but in general, you will see some increase above what you see initially in the biosolid, but it's not as substantial as what you see for the carboxylic acids. And that's because most of our precursors that degrade to things like PFAS, they have a much slower degradation rate in the soil. So you don't see that increase as much. And they're also present at much lower concentrations, the precursors to sulfonic acids. That's important to remember. And then I just want to highlight, this is what the precursors that show up in the leachate, and you do see a few of different precursors. Some of these are what are typically measured now in commercial labs. What you don't see is any of the DIPAPs because they are highly sorbed and they're transforming. They're not showing up in the leachate at all. And remember, 75% of the total fluorine mole balance associated with the PFAS we can quantify, it's coming from these DIPAPs. And they don't leach. So here's one example that I can show you right now. I'm showing you a lot of unpublished data, so I just want to be careful about that. But this is data from a dedicated land disposal site after 4 decades. And I feel like it's really interesting because you can kind of consider it like a worst case scenario. And what we see, interestingly, is in the biosolids, you see primarily precursors. That's this orangish shaded area. In the soil, and remember, this is after 4 decades of land application, you primarily see the long chain sulfonic acids and carboxylic acids, things like PFOS and PFOA and PFluoro, well, you probably don't want to hear them all, but PFNA, so all the long chain. And then in the groundwater, you might primarily see the very short chain compounds. And so what's interesting about that is, first of all, not surprising that if we keep applying biosolids, and this is dedicated land disposal where it's applied every year, that is not the normal land application for agriculture. So it's really a worst case scenario. Obviously, it can lead to accumulation of total PFAS in the soil. But what's interesting is you have this high retention of the long chain PFAS in the soil, both because we know they sorb more, but also because you have huge amounts of sorption at the air-water interface that right now, a lot of people are trying to adjust their models to account for that. So this is why even when we haven't even applied any PFAS to soil for a long time, you still get very high concentrations, again, legacy in the upper soil because of this high retention. And then you're just seeing these very short chains dominate getting into the groundwater. And really, they were primarily PFBS, that's your 4-carbon compound similar to PFOS, not one that's being regulated right now. The 6-carbon and the 5-carbon acids, 4-carbon acids. So when you think about that relative distribution, that's important because, as Jamie mentioned, drinking water is really probably our largest concern just because of very fast transfer into the body when water is being drained. So if we look at I just want to put this in perspective with where we know we have a lot of problems, and Jamie also mentioned this. So PFAS clearly do get released from biosolids. We have to recognize that. But in general, and I'm going to show you some groundwater data for Pennsylvania and Indiana in a minute, we're really not seeing PFAS from biosolids being a significant contribute to the overall exposure of PFAS to the human population. Where we see the significant impacts, and we all are very well aware, is military sites where AFFFs have been used for a long time and washed into a lot of our waterways. We see them at airports, even when from firefighting training and actually having to put out a fire. Now, of some states, limited number still, are cleaning up that foam after a fire has been put out. You know, if we had been doing this all this time, I don't think we'd be having these conversations as much. Obviously, industrial discharge, obviously land disposal of industrial wastes, not biosolids based, just like the problems that, you know, Wolverine faced, whole communities built on top of a water supply that they can't use safely. And then some biosolids have been very heavily contaminated. And sometimes we call everything biosolids, but, you know, paper mill sludge gets put into biosolids. PFAS are used in paper milling. And so when you get even a single application of paper mill sludge, I can almost guarantee you are putting on a high load of PFAS onto the soils. And then we've talked about multiple avenues for different fugitive emissions from PFAS. And based on some of the questions that we just answered, yes, it can be in rainwater. Yes, it can be, you know, people can be inhaling it in their offices, their cars, their homes. But except in a very few rare worst case scenarios, our general municipal biosolids, there isn't really evidence that they're significantly impacting our overall exposure, in my opinion. But there's a lot of research that's happening right now, a lot of funded projects. I have several, so we're going to narrow in that data gap. It's clearly a little bit of a data gap right now. I'm going to show you, this is just a snapshot, one slide snapshot of close to 100 wells that we measured in Indiana. And what I'm showing you here is land application sites and wells that we're measuring near them and not near them. And basically, you can see we're seeing very low concentrations. There was only a few that were above 10 parts per trillion. And that was the highest we saw was 35 nanogram per liter total PFAS. Almost all the PFOA and PFOS, which are bigger concerns because of bioaccumulation and longer half-lives in the body, were less than a part per trillion. We can measure that because we use quadrupole time of flight, and so we're able to measure much lower concentrations than commercial labs. We didn't see any clear trend with land-applied biosolids, but really need a much more critical data synthesis because you have to think about groundwater depth and groundwater movement and direction. So we're still working on that and just finished a large sampling campaign of river waters in the county. And that was done by my graduate student, Linda Peter. And then this work is funded from the same EPA grant that we did the work in Indiana from. And this was data out of Heather President's lab in Penn State. She's a co-PI on this project. And they've done about 115 wells in Pennsylvania. And I'm just showing you PFOA and PFOS here. And so what you can see in summary, for the 44 samples that had PFOS detected, only one was above Pennsylvania's current guideline and 7 were above EPA's guideline. And then for PFOA, we had one that was above the Pennsylvania guideline and one that was above, or 7 that was above EPA. I forgot my labels, but I combined these. So this is PFOS and this is PFOA. And in this case, Pennsylvania doesn't have very good, actually almost no guidance on putting in wells, unlike Indiana. So they can have very shallow wells. They can have poorly constructed wells, not placed in great places. And they did see much higher concentrations in general in the shallower wells. They didn't really see any clear trend with proximity to biosolids application, but as we discussed, we have to, look more carefully to understand look at depth in conjunction with groundwater flow and what aquifers being tapped into. And then we want to contrast this, which I'm not showing you here, but if we contrast it to data that the Pennsylvania Department of Environmental Protection was getting for water near places where they were concerned, industrial sites, naval bases, places where firefighting has occurred or training, they're seeing obviously much higher concentrations than anything that was being observed in rural water supplies. So I think that we know that PFAS are in biosolids. That's why we're having this conversation today. We know that they can get released from biosolids, but you asked me to kind of also capture, what does it look like relative to other exposures and just, what's a path forward? And I'm really all about controlling the sources, because I didn't show data for this either, but right now for most plants, the largest load of PFAS coming into the plants is actually coming from domestic wastewater, not from landfills. Landfill leachate has high concentration of PFAS, low volumes. Industry can have high concentration of PFAS, but might have lower volumes. And your average everyday municipal wastewater recovery facility that are getting mostly domestic waste, most of the PFAS are coming from domestic waste. We can't go in and regulate all these homes. What we can do is control the sources. And then there is a lot of ways to have adaptable plans for how we land apply. And we just have to manage the risk in a sustainable way. And as we saw, just when we needed food to feed multiple billions of people in the world, innovation helped us do that. So if we can just, be more innovative. So I just like to end with that. And I promised I would show one slide that was not PFAS because they were not able to get their microplastics person. So just in the context of everything else, this is data for pre and post Canby thermal hydrolysis process in a wastewater treatment plant to produce Class A or Class EQ biosolids. So some that, basically pathogens aren't issues, odors aren't issues, no vector attraction, metals aren't issues. And so you can see a range of several pharmaceuticals, plasticizers, antimicrobials, just a host of compounds. But what was interesting is the plasticizers, including things like BPA and other substitutes for that, they just skyrocketed after treatment just because of that high temperature, high pressure process. So when we talk about microplastics, we still have a big data gap there, but I think probably our biggest concern with microplastics will be what leaches off of them in the process in our wastewater processes. So we hypothesize that this large increase in plasticizers, which went way off our standard curves, this is pretty new data, was coming from microplastics during treatment. So with that, I will just end and thank everybody for listening. I know that was a lot of data quick, but we get a lot more out of Q&A, so we'll see how that goes.
Thank you, Linda, for that presentation. And I know we've got time for a couple of questions here, but I want to remind everyone that after the final speaker, we're going to have time for to address any of the speakers. So just because you only get maybe one or two questions now, we'll make sure that everybody gets questions at the end. So does anybody have the questions on the committee for Linda? Yeah, Rebecca.
Yeah, thank you so much, Linda. I really appreciate your point about controlling the sources and that there's a downside to not using biosolids. I've heard my friends all talking against biosolids now, and I'm like, wait, there's also reasons why you need them. So I wondered what you thought about the proposition that we had hoped to explore about drying and pyrolyzing biosolids so that you can get those benefits from the biosolids for soils without, while getting rid of the organic pollutants, including PFAS and all this other horrible stuff you just mentioned.
Yeah.
Adding to our...
Thanks, Rebecca, for asking that. I only had 20 minutes, so I took out treatment things, but we've actually done some things ourselves with pyrolysis, so I can comment on that. We actually took biosolids and pyrolyzed them at low temperature, 350 degrees, and that's low for pyrolysis, and low oxygen. And the biochar that resulted, if you ground it up, and like some people say there's no PFAS in the biochar, well, I think they're not grinding and extracting it well, because if we grind and extract it, we get a lot of PFAS back out. There's about a 30% reduction. But what's important is if you try to leach PFASs from the biochar, the only thing you see is carboxylic acids, and they're all below, like, only the shortest chain, 4 carbon and 5 carbon, are like 4 to 7 parts per trillion. The rest of them are down there at not detect. They definitely trap PFAS in a way that doesn't release them. So I think
I heard two things, that you need to get to 700 degrees or whatever, 600, definitely north of 3 by double at least. And then, but it will sop up the stuff, particularly fecal biochar will sop up the PFAS well.
Yeah, I think the low temperature was to keep as much carbon, but yes, obviously, so a lot of people are, obviously want to, optimize getting biogas, syngas, and all these things. And the problem, our challenge right now with pyrolysis, the data gap, but there's research happening in a lot of places, is what are we doing to the volatiles? Because 700 isn't going to destroy all the PFAS, 700 degrees centigrade. Same thing with incineration. But I think that it is a path forward for it's if we can make it more energy neutral than it is right now, that can be helpful. It won't change the fact that if we don't clean up things coming into our plants that go into our biosolids, if we can never land apply any of our biosolids, it's a really great resource. Biochar is not going to release nutrients and things like that. It's actually going to hold on to it's going to be different. Biochar is going to be different than biosolids. So I think we need guidance, but it can't just be banned. We absolutely need controller sources, and we need guidance, and manage, and what works in one state and in one region and one climate, it's not going to work everywhere. So I think that's where we just have to think about things.
Thank you very much. I think we better move on now. So thank you, Dr. Lee, for your presentation. Now we're going to move to our last speaker, who is Josh Sharp. Josh is a professor of the Department of Civil and Environmental Engineering, and he's the outgoing director of the Hydrologic Science and Engineering program at the Colorado School of Mines. His research is focused on understanding how microbial respiration impacts the fate of pollutants, such as trace organics, metals, and nutrients in the environment. His laboratory integrates tools from genomics, biogeochemistry, hydrology, and engineering towards a mechanistic understanding of the contaminant fate in both natural and engineered systems, with a goal of continued enhanced contaminant attenuation. A focus of this work has been on nature-based solutions such as constructed wetlands, as you see here, perhaps, riverbank filtration and aquifer recharge into sustainable water treatment infrastructure. Josh, thanks so much for being here to present your information from your lab.
All right, well, thank you for the invitation. So I think you covered my intro really well, and I'm going to begin by kind of going into my perspective and my background. So I'm a professor of environmental engineering, and I study and apply nature-based solutions. And yeah, this is a wetland that we work on in Southern California. That's a real gem. My research has a focus on biogeochemical processes and their interface with the contaminant fate in transport. Ideally, my group strives to apply this understanding towards more sustainable contaminant attenuation. As a caveat, you've heard from some amazing experts in the field. My group hasn't worked directly on the biotransformation of per and polyfluorinated compounds, and that was part of my charge. So I had a bit of reading up to do before this presentation, but we have done a lot of past work on how their presence might exert selective pressures on microbial structure and function. Of direct relevance to this larger theme, My group's research has been on the mechanisms associated with the biotransformation of emerging and trace organic compounds. PFAS, as we've heard a lot about, is one of these super pressing trace organic compounds. So here's my roadmap for the next 15 or so minutes. I'll start out with a focus on trace organics more broadly, as they provide some important lessons regardless of contaminant concern, and can provide important context for challenges and questions associated with PFAS attenuation. From there, I'll move on towards PFAS. Specifically, I'll focus on current evidence for its biotransformation, as well as other impacts that it might exert on microbial structure and function. Linda gave a really nice sort of intro to wastewater treatment and its importance in our society. And it's an amazing process. It's society's liver, if you will. We've adapted this technology over the decades to safely process municipal and industrial wastes, recover water, and produce resources like biosolids. However, it typically targets bulk organics, pathogens, and increasingly nutrients. Herein lies a problem as we learn more about the ubiquity of other anthropogenic pollutants in our environment that can come from households. One example is pharmaceuticals. These are things like caffeine, antibiotics and antivirals, hormones, and antidepressants, which are introduced into these systems daily. At present, the way these systems are managed, wastewater treatment works pretty well for degrading some compounds, like caffeine, but can be minimally effective for others. As a result, trace quantities of pharmaceuticals leave these systems in association with water and biosolids and are ubiquitous in our environment. In wastewater treatment and after environmental release, contaminant attenuation can broadly be viewed as a combination of physical processes, things like volatilization and sorption, chemical processes like photolysis and hydrolysis, and biological processes that can include biological assimilation as well as biodegradation. Many of these, such as sorption, UV, irradiation, dilution, bioaccumulation, and biodegradation, are relevant to soils. However, a caveat of biotransformation is the potential replacement of one toxic compound with an intermediate that's potentially more toxic. Linda talked a lot about this in terms of precursor formation or precursors building into PFAS and PFOA. A broader caveat, and here's a citation from one of your committee members relating to the biotransformation of the fuel oxygenate methyl butyl ether. So this caveat is something we were really concerned about in water, which was MTBE that was making its way in as a fuel additive, could be biotransformed into something that was more toxic, per butyl alcohol. We already heard about this precursor formation with PFAS and PFOA. Ideally, when we're thinking about biotransformation, we want complete mineralization. We want to convert these compounds into CO2, inorganic nitrogen, or certainly into less toxic intermediates. More broadly, when it comes to biodegradation, trace organics present a fascinating challenge. These are compounds that have toxicological concerns and are environmentally present in the parts per trillion range. For context, this is like looking for a needle in a haystack. It's less than 1 drop of water in an Olympic-sized swimming pool. I'll use arsenic as an example of a generally well accepted and common toxic pollutant in soil and water. We regulate arsenic in water at 10 micrograms per liter, or 10 parts per billion. That value wasn't chosen in part because of detection limits, and it was a huge contentious debate decades ago, or several decades ago. For trace organics, we drop down maybe 1,000 times lower into the parts per trillion range, or nanograms per liter. Because of this, it stands to reason that trace organics are unlikely to support catabolic or anabolic microbial processes, even if thermodynamically favorable. As such, they are unlikely to exert a selective pressure on microbial communities towards their metabolism. There just isn't enough to make it work. Nonetheless, and fortunately, many of these compounds do undergo biotransformation in the environment. This can be driven by detoxification. In other cases, it might be attributed to co-metabolic type processes. And this is a term I use with reservations, as it's not to say that there is no anabolic or catabolic benefits to the cells, but rather that transformation is probably not driven by this compound as a substrate. So on the left, we have an example of some work we did long ago demonstrating that a bacterial propane monooxygenase can effectively target the biotransformation of the trace disinfection byproduct N-nitrosyl dimethylamine. Surprisingly, the propane monooxygenase had a higher specificity and attenuation capability for NDMA than it did for propane, even though propane was the inducing substrate. On the right is an example of some recent work in our lab that features another member of your panel. In this paper, we provided evidence that biotransformation of the antibiotic sulfamethoxazole is associated with the expression and activity of a particulate methane monooxygenase resident in microorganisms found in these constructed wetlands. Once we understand these mechanisms, we can put on an engineering hat and think about how to construct or manage biological systems that select for favorable metabolisms that in turn could lead to more effective contaminant attenuation. In the case of NDMA, this can get as extreme as pumping propane into a bioreactor to lead to more effective degradation. On the less extreme end, a host of prior studies have explored correlations between analogous ammonia monooxygenases and the biodegradation of other trace organics in wastewater treatment. This potentially means that if we can manage wastewater treatment for the expression of ammonia oxidizing enzymes, we can increase trace organic biodegradation capability. We could make an analogous argument for engineered wetlands. Methanotrophic capacity could potentially remove more methane as a greenhouse gas and greenhouse gas forcings, while also enhancing the removal of trace organics. So, now that I've sort of laid this out, what does this all have to do with per and polyfluorinated alkyl substances? Right now, our approach to PFAS is really a mission of search and destroy. As explained by Linda, the more we look, the more we find them. As explained by Jamie, the more we explore their toxicology, and as the more we look at different sort of outcomes and vectors, the more concerned we become. Treatment at present relies on two major approaches. The first is removing it from the environment, water and soils, through separation processes like sorption-activated carbon or more selectively engineered ion exchange or other materials. It can also be accomplished through exclusion and separation by membranes or other partitioning processes. Once we've concentrated or isolated this from the environment, we then have to figure out how to destroy it. For a quick context, these are compounds that were used in firefighting foams. They're designed to be incredibly resilient under high temperature environments. As a result, we have to throw a lot of energy and muscle into their destruction, whether that be through advanced oxidation or advanced reductive processes. This has been a strong focus of several of my colleagues here at Mines, and I want to throw a shout out to Shalai Howe for some of the material on this in another slide. But here's the good news. Forever is a convenient and powerful term. It resonates. But many of these compounds are biodegradable, at least to some degree, and can break down over time, as we've already heard. In soils, there are multiple mechanisms that play a role, including sorption, volatilization, and biodegradation. However, reliable and rapid treatment applications using biodegradation are at present elusive, despite considerable effort. This is particularly true for the perchlorinated subclass, where fluorine is more complete in terms of its association with carbon or more extensive. Rates are slow, conditions are niche, and complete mineralization appears to be the exception. We also mentioned the production of PFOA, or Linda mentioned the production of PFOA intermediates earlier. This means that biotransformation of one set of PFAS or a group of PFAS compounds can lead to the formation potentially of something more toxic. Much of the literature highlights the strong carbon-fluorine bond and the difficulty in breaking this down. However, the capability to enzymatically cleave this carbon-fluorine bond was demonstrated over a half a century ago for the single fluorine contained in fluoroacetate. This is one of the few naturally forming fluorinated compounds, and it's produced in certain plants. It's pretty nasty stuff. Fluoroacetate was adopted as rat poison in the 40s and discontinued by the 80s. But this was for a single fluorinated compound. A few years ago, Eugene Min's group discovered or provided compelling evidence for the microbial degradation of branched per and some polyfluorinated carboxylic acids, as evidenced by the formation of reductive defluorination products and ultimately fluoride release. More recently, her group demonstrated microbial defluorination of an unsaturated subclass under both aerobic and anaerobic conditions. However, in these cases, degradation was associated with certain metabolisms. We don't yet know the microorganisms. It didn't actually seem like it linked directly to reductive defluorination in this case, and we don't know the enzymes that might drive these processes. This reiterates the challenge in working with trace organics since they don't directly drive metabolic selection. Peter Jaffe's group generated a lot of excitement in 2019 when they presented evidence for the microbial degradation of two of the biggies, PFAS and PFOA. This was accomplished by an autotroph that oxidizes ammonium to nitrate while reducing ferric iron, a process coined as FEAMOX. It also did so for PFAS concentrations up to 100 milligrams per liter. They recorded the production of fluoride as well as intermediates consistent with degradation during PFAS removal. More recently, they demonstrated that this metabolism could be applied towards degrading PFOA and biosolids. That said, it required really special conditions that included acclimated cultures to both FEAMOX and the past presence of PFOA. Once this occurred, though, it resulted in 50 to 80% removal over about a half a year. Again, the actual mechanism and enzymes responsible are at present elusive. A recent effort out of Texas A&M highlighted treatment synergies between sorption and degradation. And this is pretty cool when you start putting together physical, chemical, and biological processes. And they sort of engineered a system that could then hold onto and degrade PFAS. They modified lignin and cellulose, effectively really small sort of wood chip type fibers, to increase their size and their sort of activated areas, whereas functional groups. And this in turn led to increased sorption of PFAS and PFOA. This substrate also provided an active sort of place for white rot fungi to grow, and then in turn express lactases. While sorption challenged the quantification of absolute removal, they observed the formation of some presumed C6 and C7 degradation products. At the same time, they didn't report on fluoride production, and they couldn't really mass balance the removal from the system. So while they presented compelling sorption data, the biodegradation data of these challenging compounds was less complete. Larry Wackett put out a series of thought pieces over the past 2 years, highlighting the broader dilemma of perfluorinated biodegradation in particular. Here's a figure from the paper that highlights some of the attributes that an organism might theoretically need to have robust biodegradation potential. Larry drew the analogy of using fluorine in agriculture and medicine, where it's intentionally added to decrease the reactivity of design chemicals. And by doing so, it increases its lifespan as a function of these properties like bond strength and electronegativity. He also highlighted, as we heard earlier, this unique sort of third phase properties. You could call these both hydrophilic and lipophilic. And this can in turn limit transport. Unlike their chlorinated relatives, where you have made immense strides towards understanding biodegradation, there's a lack of highly fluorinated natural products that could provide evolutionary pressure to select for degradation attributes. Finally, he highlights the crux of intracellular toxicity of fluoride produced during defluorination. And remember, 1 mole of PFAS can produce a lot more than 1 mole of fluorine from a system. Here he draws on the analogy of fluoride in toothpaste for its mineral reactivity as well as its biotoxicity. It doesn't just strengthen teeth, but also limits the growth of cavity-forming microorganisms. So much like we're learning more about the toxicological impacts of PFAS and how those are driving new proposed standards, as highlighted in Jamie's talk, there's evidence that the presence of certain PFAS compounds can exert selective microbial community pressures. In a series of studies, we explored the effect of a mixed cocktail of perfluoroalkyl acids and aqueous firefighting foams, which is a total tongue twister, on reductive dechlorination by a mixed consortium. We found that it could inhibit D. helicoides growth with implications for reductive dechlorination. Effectively, the presence of one toxic compound inhibited the biodegradation of another toxic compound, in this case, chlorinated solvents. In another case, we learned that the presence of perfluoroalkyl acids results in increased EPS, extracellular polysaccharide production, and in turn, biofilm formation in a model pure culture. Gene expression was further consistent with microbial stress, suggesting larger potential impacts and a possible explanation for this microbial response. This collectively suggests that there might yet be unrealized impacts associated with the presence of PFAS on important ecosystem services provided by microorganisms. These are just two things that we found as we were actually looking at the biodegradation of other toxicants in water and trying to understand these effects. So in wrapping up, per and polyfluorinated compounds have been heralded as a generational challenge. Our concern about them grows as we learn more about their toxicity and recalcitrance. But I want to add there are a lot of anthropogenic contaminants in our environment, both organic and inorganic, that we need to be concerned about. Trace organics are an important broader example that can provide lessons for this subclass, as well as this broader challenge. Despite focused attempts by many research groups, this class of compounds, and PFAS, PFOA in particular, are highly recalcitrant, indicating that they're going to be here for the foreseeable future. Jamie mentioned timeframes of decades to even centuries. While biodegradation can play a role in their attenuation, rates are slow under environmental conditions, and enzymes and mechanisms associated with their transformation are not at present well understood. Their presence at trace quantities could help circumvent certain challenges like toxicity, but might exacerbate others like transport and natural selection. Like many other trace organics, it's unlikely that degradation will fuel metabolic processes. But it is likely that PFAS exert multiple potential effects on microorganisms, and microorganisms can, or may at least, be able to play a role in contaminant attenuation. There are a lot of unknowns in both of these domains, and just as we're finding human effects that we might not have anticipated, there is a similar potential for impacts on the health of soils and their importance to human well-being as we start to look for other endpoints and other potential effects. And with that, I'll wrap up.
Thank you very much. That
Oh,
was, yeah,
I
go
have the
ahead.
screen up.
You still have your screen up. That's okay.
Sorry about that.
No problem. Let's see if the committee has a couple of questions, then we'll go to the whole panel. Oh, Mike has a question. Thanks again, Josh. That was great.
Yeah, thank you for that. And I actually have a question, kind of an integrative question for all of three of the presenters. I'm not sure who wants to answer it, or maybe more than one. We heard about, the diet was a big source of PFAS into humans. We also heard that we had some nice presentation from Linda on how movement of PFAS through soils into water occurs, and we see that movement through there. And then, Josh, you talked about the microbial part of this. And so what I'm wondering is, from a more of a diet perspective or agricultural soils perspective, if we have PFAS going into those soils, how much of it is moving? Do we have a good handle on how much is moving into water, passing through the soil? How much is moving into the crop plants? And do we know, how much do we know about the microbial situation of biotransformation and degradation and so forth that might be going on in those agricultural soils, because we have a very particular microbial populations that occur in those agricultural soils. So a little bit of a mix from everybody.
Maybe I can start. So in terms of PFAS distribution during the life of a crop, you have more that's probably just sitting in the soil. But you'll get that certain portion that will mobilize in some places, even with well, I won't bring in wastewater effluent irrigation because there's so that's the other part of the story. But so like we said, it's mostly shorter chains that get into plants. It's very plant specific. I think right now, in terms of PFAS uptake from your everyday biosolids, which can have very high PFAS loads as a total, PFOS is going down, PFOA is going down, but we have these other things and all these precursors nobody measured before. We're seeing, it seems like the biggest concern could be PFAS going into feed crops, because then the feed crops get eaten by cattle or dairy, and they're eating a lot of it, and then it can bioaccumulate in the milk, and then milk is tends to be to our more sensitive sentinels, which Jamie can comment on, our children. So then and even in that case, like when we look at PFAS getting taken up into corn, it's very small, and that's why in Maine they've even recommended to their dairy producers like from a nutrition standpoint, they would be telling them to feed him more hay, but from just to be able to balance this challenge that they have, they want them to feed them more corn. Okay, because that's so every crop species and their specific, even with the species, genetic variations show different levels of uptake, right? Most of the data, I know I cruised right past that slide without spending much time. Most of the data that you have for crop uptake are not field studies. They're spiked pots. They're not realistic, because in those studies, you don't have what you just asked, Mike. You don't have movement below the root zone because they're intentionally watering their pots to keep everything right there in the root zone. So you get everything staying in that root zone. And so that's not fair. You have the life of a plant. A single plant can only, it's got a certain defined period. When you have different rain events, you have that movement. In terms of microbial transformation, again, that'll be very seasonal, but we have very, very busy microbes, lots of good microbial communities in agricultural soils. And so you probably can expect, if I had to venture to say, versus our column study, you're seeing actually a lot more transformation than you might see in a column study in the absence of plants, right? So, and I think that's why even the dedicated land disposal site I showed you, they were not growing crops on that land, but grass was growing just to cover it, but they're not growing crops. And even with that much application, those precursors were disappearing out of the soil. They were there in the biosolids, but once they get land applied, lots of aerobic transformation. So, I don't have 60%.
Sorry, I didn't mean to cut you off, but let's see if Jamie or Josh have any comments or answers also to Mike's question.
I don't really know what I could contribute to that particular question. There is a professor at North Carolina State University named Owen Duckworth, who has done the kinds of studies that Dr. Lee just mentioned in terms of looking at how much PFAS are in the soil and can get taken up by plants. I think he was looking at lettuce. The one interesting thing that I remember from Dr. Duckworth's work is that the level of organic amendment that he added to the soil impacted the amount of PFAS that could get taken up by the plant. So the more compost, for example, that you added, the less PFAS that got taken up by the plant. And that's about as in-depth as soil movement I could go into. But that's the one thing that I took away from his particular talk.
Thanks, Jamie. Josh?
Linda's an absolute expert in this. So, and I think she nailed it, right? Like, this is a classic fate and transport problem. We've alluded to that. So, if you have organic rich soils, you have more sorption sites, you most slow things down. You might get more microbial metabolism that might deal with some of these precursor problems. And that can limit mobility out of the system. They're funky compounds, right? They like to exist in some different places. So over time, you're going to get some dilution effects. You're going to get some degradation effects. You're going to get some immobilization effects. And the net result is going to be it's slowly going to go away, and the transport might not be overwhelmingly crazy. But really, Linda is at the forefront of this.
Well, thank you, Linda, for your comments. Are there any other questions from the committee as we move on? Or we can stay and talk about this longer. I just want to make sure that the committee has. Nick.
Hi, thanks. Great presentations. So the one thing I want to kind of interject is that a lot of the committee are going to be focusing on microbiomes of different kinds, whether they're soil or gut or whatever. And so I was trying to, in my mind, I'm not a microbiologist, but I'm trying to think, okay, so if you had your choice of microbiome or what you would create in a field or a certain type of soil management, right? That would make a certain condition that would optimize. Do you think that we're at that point, or is this just, there's not enough known?
I'll jump in and then I'll hope to dial a friend afterwards. Yeah, I mean, I'm all about building and designing systems that let microbes do their jobs as effectively as possible. The challenge right now, for the precursors, we might be able to work on breaking them down, right? So the polyfluorinated compounds are a different nugget to crack than the perfluorinated compounds. So we might be able to hit a point where we can really whittle down the overall load within a system by having sort of active microbial metabolism. It happens under both aerobic and anaerobic conditions. Originally, there was a lot of thought towards reductive defluorination, and it turns out there's some really solid aerobic precedents as well. So I think we could slowly whittle that down by having active microbial communities. There's still this crux of the sort of terminal products of these perfluorinated compounds and what sort of happens with those and their perhaps role in the larger load within the system. And from here, I might kick it over to someone else like Linda.
Yeah, so I mean, an optimal microbiome is going to first, like Josh said, it's going to break down those precursors, but that's actually not working in our benefit, but it's going to happen. It's not something we can control. It's not going to work in our benefit because most of those precursors don't get taken up by plants. They aren't mobile, but as soon as they start to break down, they are taken up by plants and they are mobile. So, but we can't control that. So, and Josh didn't comment, but we all know that even if he did comment very well, he captured where people have seen a certain amount of things, whether they had, multiple lines of evidence or not, but a lot of those scenarios to actually have to force to make them happen, even in an anaerobic, even in a wastewater treatment plant, will be quite challenging to be able to get those conditions. But I know engineers know how to scale things up. So let's just, after we get a little more evidence, maybe they can make that happen.
Thank you. More questions from the committee? Yeah, Carlotta.
Thank you. And thank you all for these excellent talks. My question is for Dr. DeWitt. And as you were presenting, several questions came to my mind. Like, for example, what is the interaction between the dose of exposure and the duration of exposure to PFAS, PFOA, PFOS? Or do they synergize with any other factors that render individuals more at risk to immunodeficiency, for example? So in your view, what should be prioritized when studying the effect of these chemicals on human health?
When we look at the maximum contaminant levels proposed by the US EPA, they're based on reference doses that are chronic reference doses. So they're designed to protect people from a lifetime of exposure to PFAS in their drinking water. And I think what we mostly have in the environment are people exposed to relatively low doses across a long period of time. And we know that those low chronic doses are the scenarios that are associated with these increased risks of the various diseases that I discussed. So the duration of exposure, and as Dr. Lee mentioned in one of her answers, the life stage of the individual being exposed are some of our biggest concerns, because we know that developing organisms are going to have different risks than fully formed adult organisms. So certainly the duration of exposure is a major consideration. There has not, to my knowledge, been as much work done on potentially sensitive subpopulations. We've already mentioned developing organisms with respect to the immune system, those who already have some immune susceptibilities because of their age, those at the beginning of life and at the end of life, or because of pre-existing conditions will have enhanced susceptibilities. But we're still really in our infancy when it comes to defining the entire toxicological understanding around this large class of compounds. Hope that answers your question.
Jamie, one thing I don't think you took time to mention, because we all kind of know it. Well, you did mention it a little bit, because I know we have at least one medical doctor on the team, is just how these compounds bind to proteins versus all the other organics that we've been worried about in the past or have had to address, and they've come and gone. They went to a very different place in the body. Do you want to comment on that? Or just give...
Yeah, I think I mentioned it a little bit when I was talking or when I was answering Nick's previous question, that PFAS, to the best of our knowledge, bind preferentially to albumin, which is a transporter protein in the blood. They can also bind to different proteins and receptors. They bind peroxisome proliferator activated receptor alpha, which is one of the receptors underlying their liver toxicity. We know they can interfere with hormone signals through binding to various receptors. They can increase and decrease levels of thyroid hormone, as well as testosterone and estrogen. And they tend to go to organs that receive a high rate of blood flow. So they tend to be in highest concentration in the liver, the kidneys, the lung, and depending on chain length, they can differentially segregate to different parts of the body, although it's still not clearly understood how the different chain lengths can influence where the compounds go in the body.
Sam. Hi.
Thank you. Yeah, hi. Thank you very much for this nice presentations. Really enjoyed these presentations. And my question is to Dr. Sharp. I was wondering about if you know what's the longevity of these compounds. We know that they are long lasting. What's the longevity of these compounds in soil? So if they are, depending on the particle size, if it's a clay soil, do you see them being more retaining in the because they are more protected? Do you think that they're going to be lasting more because they are more protected from microbial decomposition? And also I was wondering about we know that microbial communities are incredibly dynamic. The question is that is there a timeline, like how long do they need before they could break down to a more relatively simpler forms? Probably it's a naive question, but I just wanted to.
I think it's a great question. And I mean, I think your comment on sort of how soil type could influence it, gets back in some really interesting nuances between sort of net transport, locking up, absorption, as well as sort of microbial activity in the system. And I'm not aware of work that has kind of been done in that domain, but it's a really cool question. Right now, the time frame's long, right? And we have this dilemma of these polyfluorinated compounds turning into perfluorinated compounds in some cases with the precursors. So they're not going anywhere anytime soon. They are slowly going away. Like we said before, this forever term is convenient, and that's through this combination of biological, physical, and sort of chemical sort of slow attenuation within the system. But I like your perspective on how it might change within different systems, and perhaps some soils might be far more effective at sort of mitigating their effects, changing metabolism within a system, or changing sort of the transport out. But I don't have a compelling answer for it. I don't know if anyone else on the committee does.
Thank you. Yeah.
I don't think, in general, for most PFAS, clays don't play a huge role in terms of the sorptive sink, but when we talk about sorption at the air-water interface, our finer texture soils have a lot more interfacial area, so a lot more interfacial sorption, so that I guess literally the texture of soil can play a role in retention that's more than what we normally think about in all the typical kind of batch slurry studies where people are just trying to get some handle on what the partition coefficients are for different compounds. And in biosolids, carbon seems to be the big driver, but in soils, when you're now thinking about all soils, a lot of like military sites might have pretty sandy soils, and then there's different things that seem to be driving the retention, not carbon. But I know for biosolids, it's carbon. And like Jamie inferred about Scott's work, wasn't that Scott you were referring to? No. Duckweed. What's his first name?
Owen Duckworth. Yep.
I just had a brain blip. Like he saw more compost adding because you're adding more carbon.
And then Sam, one other point I recall you asked about sort of, the longevity of these compounds and how that might sort of select for microbial metabolism down the road. It's a good question again. These are mostly anthropogenic. There aren't that many natural sort of analogs that could have led to sort of, millennia or centuries of natural selection that could lead to their degradation. And we've introduced them, so that might happen. Nature finds a way. But again, I think the crux here is if it's not a viable substrate because it's present at low concentrations, thank goodness, it might not have that strong selective pressure that we need. The thermodynamics apparently work out, at least in some cases, for the degradation of these compounds. So it's possible. The biodegradation community has been banging their head on this for a decade. And inroads are being made, but there's no silver bullet.
Thank you.
Thank you. Eva. Sorry. I can't hear you. I don't know what's wrong. It says your mic is on, but
Sometimes my computer will just, of its own accord, switch to the computer microphone, which doesn't work.
oh no. Kat, have you got any suggestions?
I think if you hit the up arrow button next to the microphone, you might be able to.
I'm trying. Is it
We
working
can
now?
hear you. We can hear you.
Excellent. Well, just thinking about speeding up evolution, has anyone tried to take soil slurries and put them in the lab with these, PFAS as carbon sources and see if anything can grow and select them that way?
For sure. Yeah, that's the first approach, right? And this is how we found toluene degrading organisms, a benzene degrading organism. You throw it in and you throw a high enough concentration, milligrams per liter, whatever else, it's not toxic and things grow. Doesn't work so well in this case. So, I mean, even, the sort of the most promising work out of Yuji's group and Peter Jaffe's group, they really grew it on another condition, totally removed that, at least in Yuji's case, totally removed any sort of ancillary donors and acceptors within the system and created optimal conditions for its removal. But it's not, if only we were that lucky, right? Yeah.
It seemed too easy.
Yeah.
Any other questions from the committee?
I did want to just say one thing. I think Nick asked a question about toxicodynamics, and I don't think I fully answered his question because he asked also about other aspects of ADME, such as metabolism. I just wanted to emphasize that to the best of our knowledge, the vast majority of PFAS are not metabolized by humans. So what comes in goes out.
Which is another awesome sort of biodegradation sort of analogy, right? When we thought about degrading trace organics, we actually think a lot about human analogs. So in the case of NDMA, it's highly analogous. Its breakdown is done by CYP2E40 in the human body, which is also associated with alcohol detoxification. And so we looked around for monooxygenases that were analogous to that, and that was sort of our original hypothesis and guiding force. If you don't have that substantial human or mammalian literature on degradation, it's hard to even find inspiration. And right now, they're a recalcitrant bunch.
So I have a question. I want to respond with a comment and a question from Jamie. I know that mammalian behavior versus, amphibians and things like that, they're very different. But I just wonder if some of our lack of understanding or interpretation of human data is because we don't, for the most part, have very good data of what you were exposed to and what's in your blood. We don't have a good handle on it, outside of some of the occupational things, because in amphibians, we do see transformation in the body. And
Also,
other wildlife, we do see transformation of the PFASs going in, in our direct exposures when we're exposing them to different precursors. We can get transformation prior to that. You can put it in the environment, it can get transformed, but it's also getting, we're looking at transformation within the biota. So I just wonder if things could be happening with humans from like food digestion, because they're not getting that many. The selection out of water is small, right? There's not that many things in the water.
Well, also, so I said the vast majority, and what I should have said, the precursor compounds can be metabolized in humans, just like as in other organisms. But when they are transformed to stable end products that also are PFAS. So PFAS goes in, it could be transformed to another PFAS, and that PFAS comes out.
Thank you, Jamie.
All right, a final question from the committee to our speakers.
Nick,
Well-
you're muted.
Oh, great. Nick.
I do. I have one more question for Jamie. Okay, so what do we know about food in the gut? Is there any studies or people studying what decreases the uptake or bioavailability, or is there such a thing for PFAS? It's mostly taken in the small intestine, I imagine. But
Yeah, I think the majority are absorbed in the small intestine. I mentioned there was at least one published study done, and I probably should have looked it up during my colleagues' presentations, but I was listening to their presentation, so I didn't do that. At least one study has been done looking at food matrices and how different types of food either enhanced or slowed the movement of PFAS absorption from the gut into the body. And I can't remember the differences among those, but I'm happy to send a copy of the manuscript to the committee if you would
That would
like
be
me
great.
to do so. There is at least one drug that has been used. It's called cholestyramine. It's a bile acid sequestrant. So that has been shown in a very limited number of individuals in 2 published studies. It has been shown to decrease PFAS concentrations in the body through decreased absorption of PFAS from the gut into the body. So it doesn't enhance elimination through the blood, but it minimizes uptake. And that's a bile acid sequestrant that
now I know what to blame my high LDL content on in my blood. Okay.
That's right. That's right.
We're going to take one final question from Rebecca. We're getting close to the closing hour here.
Yeah, thank you. I think this is also for Jamie. I'm looking about the importance of like fast food packaging or other food packaging in that package of exposure pathways. It sounds so chilling that, we're actually like layering plastic onto our food. And then the other one is to do with taking it out. I heard from our Cornell PFAS person, Damian Helbling, that cyclodextrins are really good sops for, can soak up. It's a cheap molecule that's available industrially, and it's pretty good at soaking up PFAS. So, and I've seen that you can buy like a Brita filter thing in there. Like, is that something that people in high exposure areas should be actually putting into their domestic arrangements? Those are just marginalia that are crossing my mind. I figured, well, I've got you on the line.
Well, you actually bring up 2 issues that are very concerning from an environmental justice perspective, because fast food packaging does contain PFAS, although many of the major fast food chains have dedicated themselves to phasing PFAS out of their products. The problem is finding out where in the supply chain those PFAS are added and what they're in. And Brita filters are affordable for many people as opposed to a whole home reverse osmosis filter. So I think options for people, especially in lower income areas that may have been impacted by PFAS, are really important to consider, especially considering a lot of people in these communities also are going to be getting a lot of their protein from local water sources and may have homegrown fruits and vegetables for their nutritive values. So, these are folks who sometimes live in food deserts as well. So these are very important environmental justice concerns.
Thanks so much, Jamie. Thanks so much, Linda and Josh. It was great to have you this afternoon as our speakers. It was so informative. It's just really answered a lot of our questions and left us with some gaps, I think. And this is especially important. No more questions, guys. It's getting close to the closing hour.
Those are claps.
Those are claps. Oh, everybody's happy. Okay, sorry. I want to thank everybody who's online besides the committee, but just maybe watching this live also. I really want to recognize them. We will hold our next public meeting on Tuesday, June 13th, and we'll post that information on the study's website in case you want to keep track. Thank you, everyone, for a wonderful discussion and for teaching us a lot about PFAS. Hope to see you soon. Bye-bye.